Hello, Channel 2 viewers. This is Hitchcock the Sock. We are back from our long, well-deserved break from creativity. Brock the Sock, unfortunately, passed during our intermission. Brock's dying wish was to be memorialized by constant speculation as to his cause of death, as all celebrities wish to be remembered. Was it suicide, political assassination, or natural causes? Leading speculationists, government coroner autopsy, has postulated that Brock died from a very, very rare inherited metabolic disease. This disease is a fault in the efficiency of the electron transport chain. To those of you that have forgotten, the electron transport chain is the name for a series of redox reactions in which electrons are exchanged according to reduction potential, oxidizing reducing agents such as NADH into NAD+, and FADH2 into FAD. In a typical healthy cell, reactions progress from NADH through complex 1, pumping 4 protons into the cristae of the mitochondria as electrons move into coenzyme Q. Coenzyme Q, utilizing the Q pool in complex 3, pumps 2 more protons into the intermembrane space, additionally transferring the electrons into cytochrome C. Cytochrome C must utilize complex 4, also called cytochrome C oxidase, to transfer the electrons into O2, oxygen. This enzyme, importantly, utilizes heme groups forming 2 H2O, that's water, molecules, and 4 protons in the intermembrane space to help establish the proton motive force necessary for oxidative phosphorylation. This enzyme, when deficient, slows and blocks the entire process of oxidative phosphorylation and thus the generation of ATP for cellular energy. But how far does this reach? If cytochrome C oxidase is deficient, that means that cytochrome C is not being oxidized, and as such is bound up in reduced form and unable to be recycled into its oxidized form to be reduced by coenzyme Q. If coenzyme Q cannot be oxidized by cytochrome C, coenzyme Q will similarly be bound up into its reduced form. This is where it gets especially dangerous. Without the ability for coenzyme Q to continuously enter its Q pool and quench the procedurally generated radicals, these coenzyme Q radicals, while not accumulating locally as a self-resolving radical pair, can accumulate individually. Coenzyme Q is a moderately stable radical, but as ub-semiquinone, it can always transfer this single electron into something with greater reduction potential, forming a potentially dangerous radical. Past this problem, if coenzyme Q is bound up, mitochondrial NADH is bound up, causing a complete abundance of reducing agents. You might be saying right about now, what the hell are you talking about? You're speaking in a foreign language to me. What does this mean? What this all means in practical terms is that a person with cytochrome C oxidase deficiency has a lot of dangerous intermediates being built up at once and simultaneously isn't gaining the cellularly useful energy to repair, rebuild, or resolve these issues. Cells are forced to undergo homolactic fermentation as the solely effective generation of ATP, generating 2 ATP per glucose versus the oxidative phosphorylation route, which generates up to 38 ATP for glucose. Additionally, all this oxidative stress is not being resolved. Lactic acid is continuously accumulating, constantly risking lactic acidosis, stressing the liver, which will be performing the Cori cycle constantly at the cost of its own meager ATP generation. The tissues which are expected to be the most energy demanding, namely the heart and kidney tissues, with the most dedicated cellular infrastructure towards aerobic respiration, will also have the most radicals, causing abnormal tissue development and generalized dysfunction. The skeletal muscles are also weaker. Always being energy starved means less glycogen stores. The way we've described it so far may make it sound as if a deficiency in this enzyme is always immediately a death sentence, and occasionally it is. However, there's an interesting complication in question as it relates to these diseases. if it is a mitochondrial enzyme with a known mitochondrial gene encoding it called COX-1, and mitochondrial DNA is inherited solely from the mother, would that mean that either the mother must pass this on to their child, or a novel mutation must occur each time this disease occurs? And the answer to that question is, this enzyme is not solely dependent on mitochondrial DNA, and is at least partially encoded by autosomal genes, perhaps responsible for modification or other subunits of the enzyme, inherited from both parents in a recessive inheritance scheme. Thus, it seems as if metabolic pathology, again, helps science to determine exact mechanistic questions as to whether or not Brock was murdered or if he had this disease. Well, if the government's telling the truth, it would seem to suggest there would be a lot of evidence.